Alright, this is my presentation on Porin. Alright, first I want to talk about generally about proteins. Proteins have very diverse functions: transport proteins, structural proteins, proteins used for movement, very many things that a protein can do. Proteins are made of amino acids linked together by a peptide bond. They have 4 levels of structure. There's primary, which is the amino acid sequence, like what amino acids do they have and in what order. There are only 20 of them. Secondary is the conformation of the amino acids. So are they beta sheets? Are they anti-parallel beta sheets? Alpha helices? The third is the tertiary structure. That's the 3D structure of how all of those fold up together. And the quaternary is if you have multiple polypeptides linked together. Alright, specifically Porin, the background on Porin, it was discovered by Hiroshi Nikaido, and that's him right there. The location of Porin in the cell, it's in the outer membrane of the gram-negative bacteria. You can see here in the picture where the gray is, that would be a Porin molecule. It's on the outer membrane. You have the periplasm, and then outside of the cell, it's here on this outer membrane. It helps bring things from the outside of the cell into this periplasmic space. The function of Porin is a transport protein. It's embedded in the phospholipid bilayer. We just saw that. It allows things to come into the cell. It forms a water-filled pore on the external membrane into the periplasm. The diffusion by passive transport comes in. It's a channel, and it allows ions and small molecules to diffuse into the periplasm from the external environment. It is a channel, and it is passive transport, so therefore it doesn't require any energy. There is a pore in the center and also a pore in each beta barrel that make up Porin. It can be between 301 and 420 amino acids long, depending on the specific Porin. Porin can vary from bacteria to bacteria with exactly the makeup of the Porin. It has 274 solvent molecules, 3 calcium ions, 3 detergent molecules, and an unknown ligand. The primary structure of Porin, this is in the Rhodobacter bacterium. It has trypsin, chymotrypsin, glucoprotease, ASPN protease, and carboxypeptidase. The secondary structure of protein is anti-parallel beta-pleated sheet backbone. So they're not nice and straight and smooth like these parallels shown here. They're anti-parallel, so they flip back on each other. Porin was in fact the first transmembrane protein for which beta structure was established unequivocally. The tertiary structure. The tertiary structure is all these anti-parallel pleated sheets come back on each other into a loop, you can see, and make a beta barrel. So there's a pore that comes down through the center of this, if you were looking at it from the top, and it comes around in a cylinder that makes a barrel. The quaternary structure is the 3 polypeptides linked together. They're each this beta barrel. You can see that's 1 beta barrel, 2 beta barrels, 3 beta barrels linked together, and each one has a pore in the center as well as a pore in between the 3 polypeptides. Things can diffuse into each one of these pores. Porin structure, it reverses the usual pattern. Usually for beta barrel structures, the hydrophobic is on the inside and the polar hydrophilic is on the outside, but on Porins that's not the case. It's the opposite. The polar residues are on the inside and the hydrophobic nonpolar are on the outside of this kind of barrel. It helps it attract the lipids in the phospholipid bilayer of the plasma membrane, because Porin is actually a part, it's a transmembrane protein, so it is a part of the plasma membrane, and it has to attach, and that's why it has the opposite structure when it comes to the hydrophobic and hydrophilic regions of other beta barrel structures. The classification, beta barrels are classified based on stagger and the number of strands. Stagger is the inclination of the beta strands relative to the axis of the barrel. A simpler way to say that is a measure of how the edges of the cylindrical portion are held together. And then the number of strands. Porin commonly has 16 to 18 strands. In conclusion, porin is essential to bacterium. It's needed to allow diffusion of molecules into and out of the cell from the periplasmic space to the external environment. It is a transmembrane transport. It has very complicated structure, which allows for it to help the bacteria function. Okay, thank you.